In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum. How are you, dear students? I welcome you in Pakistan International School Virtual Learning System for session 2020. And this is a first year class for biology. Today in this lecture, we will see chapter number 5, which we have studied and started before. And here in this chapter and today's lecture, we will talk about viruses, which we have seen the structure and its nature, just somehow its different type of viruses in the previous lecture. So today we will see parasitic nature of virus and the life cycle of bacteriophage and its usage. Dear students, if we talk about parasitic nature of virus, so that is host specific. So viruses are very host specific, like they are parasitic in nature and highly specific to their host. Bacteriophages, if we just see different type of viruses, like bacteriophages just attack on bacteria. Tobacco mosaic viruses that are called TMV, they attack on just tobacco plants, and rabies virus in fact only on mammals. So some human viruses are specialized for particular tissues. For example, HIV is specialized for T cells, T lymphocytes, or those are called white blood cells. And polioviruses reproduces in spinal nerves. Some viruses infect only on liver cells. Human cold viruses that affect on the lining of respiratory tract, and viruses have protein spikes on the surfaces which help them to attach with the specific receptors. So specificity of attachment determines the host range, what type of receptors they are having to attach with the host. The next nature of the virus, that they must require host cells to complete their life cycle, as we know that they are living in the living organisms. They are non-living without the host or out of their body. So they parasitize in order to complete their life cycle, because viruses are obligate intracellular parasites, which mean they cannot multiply outside of the body or of the living cells. So viruses infects all sorts of cells, like bacterial cells to human cells, plants, as well as all different type of animals also. Viruses lack, we can say, the enzyme, metabolic enzymes and ribosomes, so for making proteins. So they need host for all these purposes, for reproduction and for replication of their genetic material. Then survival of viruses inside the host cell, that is very important and complicated as well, because it needs some of the mechanisms. First of all, it blocks the complement activation, means the enzymes of activation. It blocks all the enzymes of activation of the host, like enzymes. It blocks the enzymes of the host. Number two, that it using complement receptors to enter B lymphocytes, because if we just talk about HIV, that is very lethal. We can say the virus which attacks on T lymphocytes. So it blocks the entry into, like when it just enters or just attach with T lymphocytes. So it using its complement receptors, like as we have seen that viruses is having spikes on it like this. So these are the specific receptors which help them to bind on the T lymphocytes. So these are called complement receptors, which are used to bind on T lymphocytes that are compatible or that are attached with the um, receptors which are present on T lymphocytes. The next one, like inhibiting interferon induced antiviral response. So these and the viruses in what they do, they inhibit the interferon response. Interferon is the antiviral protein which is released by host cell, that is also called signaling protein, and that just give messages to other cells to get ready for attacking on the viruses which is coming inside. But this virus, it inhibits the interferon antiviral response, blocking production of cytokines or response of cytokines. Cytokines actually, they are also proteins for signaling to the cells for attacking on the virus. So, but they are also blocked by the viruses. Suppressing major histocompatibility complex, that is called M H and C, major histocompatibility complex, that are the proteins to recognize foreign substances in human, and that is present in human leukocyte antigens. These are also called human leukocyte antigens. So they are like immune response against the viruses, but they also suppress this major histocompatibility complex. Then reducing beta cells activation, they also reduce the beta cells, B cells activation, like T lymphocytes also signal the B cells and for attacking the viruses, but they also reduce the B cells activation when they just destroy T lymphocytes. So changing their own genetic constitution so rapidly that vaccines or antibiotics of host against them become ineffective. So we can see that we cannot use antibiotics for viruses because they reduce or they change their genetic material. So we can say the viruses are mutant and they can change their genetic material according to the situations from outside. Viruses, the next very important nature of viruses is that they can pass unfavorable condition outside the host. They are very small, so other organisms cannot attack on that and cannot threaten and they are not threatened by other organisms, and their only concern would be the pH and temperature. If pH and temperature is going to be changed, so the protein coat outside their like core, that is protein capsid, they can be destroyed and they can destroy easily. So they can live and undergo dormant period for longer and longer period of time because they are non-living components outside the host. So non-enveloped viruses are also, they can infect, survive for longer period of times, up to several days, whereas enveloped viruses survive for shorter period of time because they are having lipoprotein on that, and enveloped viruses rely on the proteins on the surfaces of the membrane to attach the host cell. So this envelope is generally sensitive to degradation of sunlight and normal cleaning procedures like Antiseptics and antiviral agents, they can destroy these proteins, and the envelope can get ruptured and break down, because on that there are spikes of glycoproteins that can be damaged with using of cleaning procedures. Now we will see bacteriophage and its life cycle. As we know that viruses that infect bacteria, they are called bacteriophages. So they occur widely in nature in close association with bacteria, and rarely isolated from feces, sewage and other natural sources, because bacteria are present in those places and they attack on bacteria. So there are two types of life cycles of bacteriophages. Number one is virulent, that is lethal for bacteria, and bacteria can destroy in that die, that is called lytic cycle. Then the other one is temperate, or that is called lysogenic cycle. So let's go through both of them. Lytic development, if we see the first process, that is called infection stage, and this cycle is also called master slave relationship. Master slave relationship means the master is the bacteriophage and slave is the bacteria, because this bacterial DNA that is in the red one, that is hijacked completely with the DNA or that genome of virus. So phage attaches to the bacterium in infection stage. Then there is DNA injection, in which phage injects DNA into the bacterium. Then in early infection, phage DNA replication starts, and in late infection, the head, tails, and fibers are made with the help of this DNA of that is of bacteria. So then later on, DNA is packaged into heads, tails, and become attached. And after that, when there are lots of viruses are produced, in that the cell is broken down and progeny of virus is released. So because here the bacterial cell dies, that's why that is called lytic cycle, and that is virulent also. So here we can see absorption of bacteriophage to the cell wall, that is called adsorption also, and protein on viral surface attached to the specific receptors on the host cell surface. Then we can see the viral genome that is penetrated inside the cytoplasm, and this step is known as entry, and that can be DNA or RNA. Now replication and maturation of a virus, when this genome is entered, then it translated by the host cell, with combining with the host cell. We are having here viral genome. The protein parts are produced and then assembled. The next assembly and the lysis is done when the lots of viruses are gets assembled and then released out from the cell. So the bacterial cell did lysis, do lysis at the time. So the first step is adsorption, then entry, then replication, then assembly, and then lysis. So that can also be just changes into like synthesis process, assembly process, maturation process, and lysis or a release process. Now we will see lysogenic cycle. In lysogenic cycle, the same cycle is going on in the entry and release of genome into the bacterial cell. Then this bacterial cell with the phage DNA circularizes, or it can go into the lytic cycle, or it can go into the lysogenic cycle. So what happened here, the genome of virus get penetrated or get the part become the part of DNA of bacteria, so it gets prophage. So this DNA integrates into the bacterial chromosome, become prophage, and when the bacterial genome gets replicated, this viral genome also, or prophage also transmitting into the daughter cells, or just with this DNA of bacteria, or replication, or fission, we can say the binary fission of bacteria that also become divided. So it will not produce its all other parts, but it will replicate and divide with each cell of bacteria. So many cell division produce a colony of bacteria, but that is infected with a prophage. So occasionally a prophage exists in bacterial chromosomes and initiating the process again if it is required under unfavorable conditions. What happened, this gets into the virus gets into the bacteria, and with bacteria that goes into lysogenic cycle, and but in favorable conditions, so lytic cycle is done and lots of viruses are released. That is called host guest relationship, in which bacteria is host and viral DNA is guest. And here we can say that is master slave relationship. Here the bacteria is slave and virus is master. Now, if you see this summary of that one, first absorption is occurred, then entry, then integration means formation of provirus of prophage, then spontaneous induction, the viral DNA from the host, then again replication, assembly, lysis, and uh, release of that viruses is done from the lytic cycle. Now we'll see some uses of bacteriophages in genetic engineering. So the natural predators of bacteria can be used efficiently in modern biotechnology and propose alternatives to antibiotics. Antibiotics, actually, bacterial strains are get resistant to antibiotics. Now these antibiotics are replaced by these bacteriophages, and these phages can be used as biocontrol agents in agriculture and petroleum industries as well. So very important use of that is genomic library. We can bacterial plasmids are utilized for that purpose, but now phage DNA has also been used as vectors in genetic engineering. So there is the development of genomic library where there are lots of collection of genomic like a genome of that viruses of bacteriophages, and there are multiple copies of that, and we can utilize it for our purpose. Phage therapy, that is the application of genetic engineered phages that can kill pathogens, bacteria, like we release that bacteriophages on the bacteria, and they digest or undergo through the lytic processes. So that is conventional antibiotic therapy, and like that is also called the phage treatment to kill bacterial pathogens while avoiding harming of normal bacterial flora in the body. So because they are host specific, so they go just on to the specific bacteria. The next was antibacterial nanomedicines. So they are used for many years for the tools for different purposes, but now antibacterial nanomedicines are used, and they entered in the body in the medicines form, and they can be formulated as a targeted drug delivery vehicles, like they just attack on the required cells, on the targeted cells, to kill not only bacteria but some other like for cancer, for some other diseases also that can be used. So that is called novel drug delivery. So that is the new procedure which is going to be used nowadays in the next coming future, and that is very, very helpful. Now we very little bacteriophages we can use for our purpose in the form of genetic engineered conditions. So dear students, that's it for today. Inshallah, we will continue our next topic about some of the diseases and HIV and its infestation and its spreading and transmission. Till then, wish you all the best. Allah Hafiz.